I'm Cayla Martin, and I would also like to introduce you to my birds because they may be actively contributing to my presentation, so I apologize in advance. But I am really excited to share the research that I have been conducting at the Rozel Seeps. Oh, there we go. But first, I would like to thank everybody who made this experience possible for me, namely Great Salt Lake Institute with Bonnie, David, and Jamie. Thank you for putting up with my crazy obsession and weird ideas with the Seeps. Also, this would not have been possible without the generous donors. Being able to constantly go out there and monitor and collect new information and data is really important. And then also really importantly is previous students' contribution. Kara Kornheiser, she started this entire research project, and I'm really grateful for that because ever since then, she's graduated and I've been able to continue and answer some of the questions that she has asked. And I see that Greg McDonald is also on the call, and I know that he worked with Kara, so that's really awesome. And then also Mary, who has also graduated and who provided an amazing platform for me. So I'm going to be talking about two different projects that I have been conducting. The first one is about the Rozel Point tar seeps and their impact on the local biology. And the second one is the potential microbial diversity. So the tar seeps are located in the north arm of Great Salt Lake, just below Spiral Jetty, as you can see there with that red dot. And I think the best way for me to kind of explain the tar seeps is to share my first experience when I went out there. And basically you arrive at this completely desolate, quiet place where you just have these mountains and the pink lake surrounding you. And I immediately knew that this is a place that I would love to know everything about, and I'm completely hooked. And so while you are walking to the seeps, you definitely first smell them before you see them. And with this, I got this huge whiff of decay and asphalt. And then when you arrive at the seeps, there's this incredible natural phenomenon that is occurring. Namely, you have gas and oil that migrate through cracks and fissures and then pull out on the top of the surface. And this can be seen in different ways. For example, in this image, you see the tar kind of blanketing over the sand. And then sometimes when the tar connects with water, you get, in my opinion, this really artistic interaction. And again, this is just like, I fell in love. I thought this was so cool and I wanted to know everything that I could. And then sometimes you also get volcanoes where the tar pulls up over the surface and then hardens, and you get these like harder vents on the outside, and it's a little bit more sticky on the inside. And these come in different shapes and sizes. You have big ones that can sometimes fill up with water when it's colder, and then you have smaller ones. And so it turns out that these seeps are located along a migratory stop for many bird species, particularly the American white pelican. I took this photo in the beginning of the summer, and it's just really cool because you hear the pelicans flying above you and the wind whistles through their wings, and it's really, really cool. But on the flip side of that, there's also evidence, besides the fact that you see the pelicans flying over, but you also see evidence that the pelicans are actually dying. So here we have a pelican skull with its beak that you see dotted around the seeps. And there are a lot of pelicans that actually get entrapped in the seeps themselves. And so when Kara went out there at first and saw all these pelicans, of course, she had so many questions. For example, what animals are getting entrapped in these seeps? Is it just American white pelicans, or are there different kinds of animals, just birds or other species entirely? What are the effects of temperature? How does the temperature of the surrounding environment affect the temperature of the tar, and therefore how sticky it is? And are there other animals that are scavenging on these pelicans? And so Cayla set up cameras around, I'm not sure if you can see my mouse, my cursor, but this is on the left-hand side, you see the biggest seep, and there are these little white dots, and those are all pelicans that have been previously entrapped. In the middle, you see...
Yeah, I can see your cursor, by the way.
Oh, great. Thank you. In the middle, you see this like little seep, which is the volcano seep. So that's where you see all of the different volcano mounds. And then on the right-hand side, we named this peli seep because it kind of is in the shape of a pelican. And this one is really interesting because it has the largest range of diversity of different animals that visited that seep. And so there are a total of 8 cameras set up all around these seeps. As you can see here, this camera is motion activated. So when something comes within its range, it triggers the camera and the camera takes multiple pictures. We also have the camera set on time lapse, which has been super interesting, which I'll get into later, but we're able to see the movement of the tar. And so with these cameras, we've been able to see really, really cool things, such as these pelicans. Notice the time of day, it's in the morning. And so perhaps the tar is not as sticky because these 2 pelicans did not get entrapped. And unfortunately, that is not always the case. This pelican, again, notice the time of day, it's later on in the day. The tar might be a little bit hotter and therefore stickier. And so this pelican actually did get entrapped. And I kind of just want to pause here for a bit. This was really strange for me because obviously I had never done a research project monitoring animals, but never mind monitoring the death of animals. And so I had this like mixed feelings of, I was really excited to be able to learn more about the seeps and this strange environment. But on the flip side of that, I'm basically counting the number of deaths. And so that was kind of interesting. Pelicans were not the only animals that visited the seeps. Coyotes came, such as this one. Again, notice the time of night. The tar may not be as sticky. Also, it's later on in the year. But so this was taken in 2018, and actually last summer, a coyote was entrapped, and we captured it that this younger coyote decided to scavenge during the day when the tar was perhaps a little bit stickier. And it sat down to feed on this pelican to the left of it, and it never got up. And again, weird feelings of emotions because this is kind of like a dog. And so that was really interesting. But what has been super cool for me is like every time I visit the seep, I'm able to take pictures of the coyote and watch this decomposition in real time. And essentially the fossilization of these animals, because the tar is able to preserve them so well. And that's important because we're able to look at the natural history of these Rozel tar points and get a better picture and understanding. And then we have this little guy, his name's Jerry. He's he was the mascot for the Seeps because he used to visit the Seeps every night around about the same time and scavenge or feed off of the water boatman bugs that got entrapped. But again, unfortunately, he himself got entrapped. And this just brings up a lot of questions, same as what Cayla asked. Why are these animals getting entrapped, and what is the relationship between temperature and number of entrapments? And as you can see here, we have the months on the y-axis and then the temperature in degrees Celsius. And the temperature is surprisingly the highest around August. And that is when we have had the most amount of entrapments. As of with this year, we had 12 new pelican entrapments and 3 new unidentified birds. All seen in August. So that's really interesting. And I guess the question is, what does this mean for future implications? I think in a climate changing world, when we are seeing increases in temperature, I believe that there might be a longer time period when the tar is really sticky, and therefore perhaps increasing the amount of entrapments or number of entrapments, because there is a longer period of time that animals could get entrapped. And so I see I might be running out of time. So I'm gonna move away from this death.
You're good on time.
Okay, good. So then back to the future implications. I think it's really important to think about the effects of rising temperatures in a changing world, and things that and the effects that it has on these natural environments, and how that can really impact the local biology. And so while it's really interesting to study all this death at the tar, recently I have been researching possible life in the tar. And so I'm going to play this video. This was captured at the volcano Seep, so the middle one. And basically this is just one of the volcanoes, and this bubble comes up and completely deflates again in a matter of minutes. And when I saw this with Mary, who has graduated, we just went crazy. We were just so confused and excited by this. What is causing this, and what does it mean? And so we started noticing bubbles, lots of little bubbles scattered along the seeps. And so Mary did a lot of research into this and found that very possibly these bubbles might be caused by burping bacteria. And so that's kind of a weird concept. But what Mary found was that there, at the La Brea tar pits in California, some scientists sampled the tar and extracted DNA and found that there was just this incredible microbial diversity living in the tar. This means that these organisms are super specialized and have evolved to be able to withstand such extreme conditions. And so this is a image taken from that paper where you see the different species. And some of them had never been described before. And so Mary graduated, and we're roommates. And so we often just like have tons of conversations about this all the time, and how we're going to find out if our very own tar pits have life in itself, when we're so used to just seeing death. And so this is kind of interesting because tar is so weird. You know, the DNA extraction kits are all created for soil samples. And so it's been really cool because I've been able to be creative and think of unique ways in trying to sample the tar, as you can see here. And so this involves trying to be as sterile as possible, sampling the tar and immediately trying to keep it cold, keep it frozen so it doesn't melt. And so in the lab, I freeze it with liquid nitrogen and try and grind it up into a really fine texture so that I'm able to use the soil DNA extraction kit to try and extract DNA from the tar, which is really difficult. And so I have to do quite a few subsamples in order to get enough DNA in a sample. And so that's where I currently am. My next step would be to amplify 16S RNA genes using PCR. And then this is my favorite thing. We're going to send that to a guy called Mr. DNA in Texas, which I think is hilarious. And hopefully we will be able to find that, you know, this is an incredible ecosystem that has a ton of life that you would not expect to be in such a harsh environment. And so those are the two projects that I'm currently doing. And again, I just want to say thank you so much to everyone at Great Salt Lake and previous students and donors to make this possible, because this has been the most amazing experience for me. Any questions?
Thank you, Cayla. We have a couple minutes for questions.
Hi, Cayla. This is Greg.
Hi, Greg.
Great presentation. I enjoyed it very much.
Thank you.
Did you think about sampling some of the tar next to one of the dead animals?
Definitely. So I've been taking 3 or 4 samples of the tar right next to dead animals, and then as a sort of control about like a couple of meters away. And so I'm assuming that that might, that will have an impact.
I look forward to hearing the results from Mr. DNA.
Thank you.
What was the rodent that got stuck? Were you able to identify it?
Let me go back to the picture quickly. The rodent, Jerry the mouse, I'm not really sure what particular rodent it is. But let me quickly go back. So this is the guy. And I'm not really sure particularly which rodent.
Okay, I'm used to looking at only the bones, so when you put the skin on, it makes it confusing for me.
I could definitely organize you some bones if you would like.
All right. I'm just wondering, you said it was out foraging on insects?
Yeah, water boatman.
I'm wondering if it might be a grasshopper mouse then, because they feed on insects.
That's awesome.
Well, I'll let somebody else ask questions.
Wonderful. We have time for one more question. Preston has one.
Yeah, so a really interesting presentation. Thank you very much. And I've been to the tar seeps a number of times myself. Will your techniques identify only bacterial DNA, or will they also pick up like viral DNA or RNA?
So that's a really great question. At this point in time, I'm focusing on bacterial DNA because the paper from the La Brea tar pits had most of their organisms or species, they were bacteria. So, but this is definitely an ongoing research, and I'll definitely want to branch out to that too. But at this point, I think just like being able to extract it and freeze it is, yeah, it's a little bit tricky.
And once you get your methods down, you can always change primers and look for other things. It's awesome. Awesome. Okay.